Dear students, welcome to KPS Bio Tutorials. In the previous episode of biochemistry classes, we studied about the glycolysis, in which we discussed that the 6-carbon compound glucose is converted into two molecules of 3-carbon compound, namely pyruvic acid. We also studied that this pyruvic acid has many fates. In skeletal muscles, it may convert into lactic acid, which is called lactate fermentation. In yeast cells, it undergo ethanol fermentation. And in aerobic cells, the pyruvic acid is converted into acetyl coenzyme A. Then the acetyl coenzyme A enter the Krebs cycle and undergo complete oxidation in the presence of oxygen. And in today's episode, I would like to start with Krebs cycle. And today's episode is especially discussing about formation of acetyl coenzyme A from pyruvic acid. After glycolysis, the pyruvic acid is formed. The pyruvic acid will be converted into acetyl coenzyme A. This acetyl coenzyme A then enter the Krebs cycle, or which is called citric acid cycle. The citric acid cycle, also called tricarboxylic acid cycle or TCA cycle or Krebs cycle, after its discoverer Hans Krebs. Hans Krebs described the Krebs cycle. He discovered the Krebs cycle, thereby citric acid cycle, also called Krebs cycle. The citric acid cycle involves the conversion of pyruvic acid or pyruvate with 3 carbon into acetyl group, into an acetyl group with 2 carbon. Here 3 carbon pyruvic acid is converted into acetyl group with 2 carbon. That means here 1 carbon atom is released in the form of carbon dioxide. And this acetyl group means it is the CH3CO group. This acetyl group is then transferred into a coenzyme A to form acetyl coenzyme A. Then this acetyl coenzyme A enter into citric acid cycle. The conversion of pyruvic acid into acetyl group and its transfer into coenzyme A to form acetyl coenzyme A. All these reactions are catalyzed by an enzyme complex, namely pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex. It is an oxidative decarboxylation. Here 1 carbon dioxide is eliminated and also NADH is also formed. So that means it is an oxidation as well as decarboxylation. Thereby the reaction is called oxidative decarboxylation. Then let us discuss how the acetyl coenzyme A is produced. Acetyl coenzyme A means it is the activated form of acetate, because as the acetyl group or CH3CO group is attached to coenzyme A, then it gets a high energy bond and thereby it is said to be activated acetate. The pyruvate derived from glucose and other sugars by glycolysis is oxidized into acetyl coenzyme A and carbon dioxide by the pyruvate dehydrogenase complex. So the enzyme complex catalyzing the formation of acetyl coenzyme A from pyruvic acid is said to be pyruvate dehydrogenase complex. A cluster of enzymes. This pyruvate dehydrogenase complex is a cluster of enzymes with multiple copies of each of three enzymes. There are three enzymes in this complex, and each of these three enzymes are present in multiple copies. The number of copies may vary from species to species, and they are located in the mitochondria of eukaryotic cells and in the cytosol in the case of prokaryotes. Now let us examine the structure of coenzyme A. Here the coenzyme A mainly consists of three parts. One is a 3-phosphoadenosine diphosphate. 3-phosphoadenosine diphosphate. Here you can see there is a ribose sugar to which an adenine compound is attached at the first carbon atom, and at the third carbon atom of the ribose sugar there is a phosphate, and in the fifth carbon atom there is a diphosphate. So totally three phosphates are there in this adenine nucleotide, but two of them are present at the 5' carbon and one at 3' carbon atom. Thereby, though it is with a 3-phosphate, it is not an ATP. It is said to be 3'-phosphoadenosine diphosphate. This is the one of the components of coenzyme A. Then there is another a vitamin partner, namely pantothenic acid. It is a member in the vitamin B complex. This pantothenic acid join with the phosphate diphosphate of the adenine nucleotide. Then after that there is another compound, namely beta mercaptoethylamine. Beta mercaptoethylamine. So these three compounds form a complex, namely coenzyme A. To this compound, here there is an SH group at the beta mercaptoethylamine. There is an SH group. This is called reactive thiol group. To this SH group, the acetyl part will be added to the coenzyme A. So the acetyl part, that is CH3CO part, will join with the SH group of beta mercaptoethylamine of coenzyme A, and thereby the acetyl coenzyme A will be formed. You can see here the structure of acetyl coenzyme A. Here this is the acetyl part which was derived from the pyruvic acid. This acetyl by the oxidative decarboxylation, this acetyl part join with the SH group of beta mercaptoethylamine to form acetyl coenzyme A. Now let us examine the components of pyruvate dehydrogenase complex. I told you there are three enzymes in this complex. These are these three enzymes. First one, pyruvate dehydrogenase, which is said to be E1 or enzyme 1, pyruvate dehydrogenase. Then second one, dihydrolipoyl transacetylase. Dihydrolipoyl transacetylase or enzyme 2, E2. Thirdly, dihydrolipoyl dehydrogenase or enzyme 3, E3. These are the three enzymes present in the pyruvate dehydrogenase complex. Each of these three enzymes are present in multiple copies, and their number of copies of each enzyme, and therefore the size of the complex varies among species. The number of copies of these three enzymes are varying in different species, thereby the size of the pyruvate dehydrogenase complex will be varying among the species. So let us examine what are the steps of acetyl coenzyme A formation from pyruvate. Through 5 steps, the acetyl coenzyme A is formed from the pyruvic acid. First step, pyruvate react with the bound thiamine pyrophosphate of pyruvate dehydrogenase or enzyme 1 undergoing decarboxylation to hydroxyethyl derivative. Here the pyruvic acid, in the picture you can see, this is the pyruvic acid CH3COCOOH. This CH3COCOOH or pyruvic acid, which react with the bound thiamine pyrophosphate. This is thiamine, this thiamine pyrophosphate, which is a cofactor for the enzyme 1, that is pyruvate dehydrogenase. So it reacts with the thiamine pyrophosphate present in the pyruvate dehydrogenase enzyme or enzyme 1, and it undergo decarboxylation. The carbon dioxide, a carbon dioxide molecule is eliminated from the pyruvic acid. The COOH group of pyruvic acid is eliminated, or it eliminated in the form of carbon dioxide. Thereby COO part of the COOH will be eliminated as carbon dioxide. Thereby one carbon atom is reduced from the pyruvic acid and it is get attached with the thiamine pyrophosphate, and the hydrogen atom of the COOH group is get delivered into the second carbon atom, that is thereby it is given to the oxygen CO group, thereby it becomes COH, and it is also given one more hydrogen atom from the thiamine pyrophosphate, thereby CHOH group will be formed. And now the compound CH3CHOH group will be attached into thiamine pyrophosphate. This compound is now called hydroxyethyl group. Hydroxyethyl group. Once again repeat. The thiamine pyrophosphate is the cofactor for the enzyme 1 or pyruvate dehydrogenase. The pyruvic acid, that is CH3COCOOH, undergo condensation with thiamine pyrophosphate. During that reaction, carbon dioxide is eliminated from the COOH group, and the CO group is get protonated into CHOH, and thereby the hydroxyethyl group is formed. This hydroxyethyl group will be attached with the thiamine pyrophosphate. This is the first step. In second step, transfer of acetyl group from TPP to the oxidized form of lipoyl lysyl or lipomide group of the core enzyme by the enzyme dihydrolipoyl transacetylase E2 to form the acetyl lipomide. Here you can see the thiamine pyrophosphate is now its hydroxyethyl group. This from thiamine pyrophosphate, an acetyl group is transferred from the thiamine pyrophosphate into another compound, namely lipomide. This is the lipomide. It is a part of the enzyme 2. It's a cofactor for enzyme 2, lipomide. And this lipomide, which is in normally it will be in the oxidized state with the ANSS disulfide bond, and the enzyme namely dihydrolipoyl transacetylase or E2 enzyme dihydrolipoyl transacetylase enzyme will transfer the acetyl group from thiamine pyrophosphate into lipomide. And the acetyl group, that is CH3CO group, form a bond with the sulfur atom in the disulfide bond of the lipomide, and thereby the disulfide bond will be broken, and it then form a bond with the CO group of the CH3CO group, and the another sulfur atom is get protonated to form SH group. Thereby a compound like this is formed, which is called acyl lipoyl lysine. It is called acyl lipoyl lysine or acetyl lipomide. It's also called acetyl lipomide. This acetyl lipomide in next step undergo transesterification with coenzyme A. That means the same enzyme, that is enzyme 2 dihydrolipoyl transacetylase, will transfer the acetyl group from this acetyl lipomide into coenzyme A. Thereby CH3CO group from the lipomide is then transferred into coenzyme A. You know, in coenzyme A there is a SH group, and to that SH group the CH3CO group will be bonded. Thereby the acetyl coenzyme A is now formed. This is the acetyl coenzyme A. And then what happens to lipomide? Lipomide is reduced into lipoyl lysine or reduced form of lipomide. So here there is 2 SH groups. Normally it will be in the oxidized state with SS disulfide bond, but after the transfer of acetyl group from the acetyl lipomide into coenzyme A, it is get reduced into SH with 2 SH groups. That is called reduced form of lipomide. Then in the fourth step, an enzyme namely dihydrolipoyl dehydrogenase, the third enzyme, enzyme 3 dihydrolipoyl dehydrogenase, promote the transfer of 2 hydrogen atoms from the reduced lipoyl groups of enzyme 2 to the FAD prosthetic group of the enzyme 3. This dihydrolipoyl dehydrogenase enzyme is having a prosthetic group namely FAD. This FAD will take up the 2 hydrogen atoms from the reduced lipomide. Thereby the hydrogen atom from the SH groups is taken by the FAD. Thereby FAD become reduced into FADH2. This reaction is catalyzed by dihydrolipoyl dehydrogenase, the third enzyme in the pyruvate dehydrogenase complex. And thereby what happens? The reduced lipomide is then again oxidized into its formal form, that is oxidized lipomide, and in this way the cycle may continue. It can take up another acetyl group from the thiamine pyrophosphate, and in this way the reaction is continued. And in the fifth step, the reduced FADH2 of E3 transfers a hydride ion into NAD, forming NADH. Here the FADH2, the reduced FAD, that is FADH2, donate its electrons and hydrogen atom to NAD plus, thereby it is reduced into NADH plus H plus. This is the fifth step in the formation of acetyl coenzyme A. Actually, the acetyl coenzyme A is formed in the third step itself. Fourth step is the regeneration of oxidized form of lipomide, and in the fifth step It is the formation of NADH from the FADH2. So at the end of the reaction, at the end of these 5 steps, from one pyruvic acid, one acetyl coenzyme will be formed, also one NADH also formed. So by the glycolysis, one glucose molecule give 2 pyruvic acid. So from 2 pyruvic acid, 2 acetyl coenzyme will be formed, also 2 NADH also formed. So this is the overall reaction catalyzed by the pyruvate dehydrogenase complex. This is one pyruvic acid, CH3COCOOH. It is in the ionized form, thereby it is called pyruvate. Pyruvic acid means it is the non-ionized form of pyruvate, that is CH3COCOOH. Then see, this pyruvic acid undergo oxidative decarboxylation, and as a result there is a decarboxylation, that means elimination of one carbon dioxide. Normally the carbon dioxide is eliminated from the COOH group, and from the pyruvic acid one carbon dioxide is eliminated. And also one NAD take up the hydrogen atom, and NAD become NADH plus H plus, and as a result of the whole reaction catalyzed by pyruvate dehydrogenase complex, finally the acetyl coenzyme is formed. First the acetyl part is formed, then the acetyl part from thiamine pyrophosphate is get transformed into lipoate. From the lipomate it is then transferred into coenzyme A, and thereby the acetyl coenzyme is formed. Carbon dioxide is released, also one NADH also released. So this is the here the delta G0, the net energy change is minus 33.4 kilojoule per molecule of pyruvic acid. So I hope all of you understood this topic very well. Thank you for listening.